The overall goal of the following experiment is to measure vacuolar and cytosolic pH in living yeast cells and to observe how pH changes with alterations in extracellular conditions. This is achieved by introducing pH-sensitive fluorescent probes to the vacuole and cytosol of yeast cells. The dye BCECF-AM is introduced to the cells in order to measure vacuolar pH, and a pH-sensitive GFP, fluorin, is expressed from a plasmid in the cells in order to measure cytosolic pH. As a second step, fluorescently labeled cells are washed and resuspended at high density in growth medium lacking glucose. Aliquots of this suspension are distributed to calibration buffers of different pH to create the calibration curve. Next, another portion of the suspension is diluted in a weak buffer, and the fluorescence of the suspension is continuously monitored in a fluorimeter before and after addition of glucose or other additives in order to determine vacuolar or cytosolic pH responses in vivo. Results are obtained that show time and condition-dependent pH conditions in the experimental sample based on ratiometric fluorescence ratios calibrated from the calibration samples.
I'm Dr. Patty Kane, Professor of Biochemistry and Molecular Biology at SUNY Upstate Medical University. This method can help answer key questions in the field of pH regulation.